Welcome back to The Explainer. Today, we're diving into some seriously amazing source material about the ultimate biological machines keeping us alive right this second. We're talking about the microscopic engines running constantly inside your cells. Honestly, without them, biology as we know it just wouldn't happen.
So, picture this life or death scenario. What if the essential chemical reactions in your body, stuff like digesting your lunch, copying your DNA, making energy, what if they were just fundamentally too slow to keep you alive? Well, according to the science, without some kind of molecular intervention, our metabolism would drag so much that a single reaction could literally take millions of years. Millions!
Right, so enter our hero molecule, the enzyme. Technically speaking, it's a biological molecule that supercharges the rate of a chemical reaction by lowering the activation energy needed for it to kick off. Think of activation energy like this massive, towering wall a reaction has to climb over. Enzymes, they act as biological catalysts and essentially tunnel right through that wall. It's an elegant, lightning-fast solution to that whole millions of years problem.
Okay, let's dive into the roadmap for today. We're covering 1. Speed machines. 2. Anatomy of an enzyme. 3. The perfect fit. 4. When enzymes break down. And finally, 5. The future of biotechnology.
All right, Section 1, Nature's Ultimate Speed Machines. Let's see these biological catalysts in action.
So the way these machines actually operate boils down to a pretty straightforward equation. An enzyme bumps into a specific molecule, we call that a substrate. They lock together, forming an enzyme-substrate complex. The reaction sparks and bam, the substrate transforms into a completely new product. And the best part? The enzyme comes out the other side totally unchanged, completely ready to do it all over again. And while most of these are complex proteins, the sources point out that certain RNA molecules, called ribozymes, can actually act as catalysts too. Now, since we're dealing with speed machines, scientists obviously need a way to track just how fast they can go, right? That's where enzyme kinetics comes in. The two heavy hitters here are Vmax and Km. Vmax is exactly what it sounds like, the absolute maximum reaction speed. It's top gear, happening when every single enzyme is completely saturated with a substrate. Then you have Km, or the Michaelis constant, which basically measures chemical magnetism, or affinity. It tells us how eager the enzyme is to grab its target, and counterintuitively, a lower Km actually means a higher affinity.
Moving on to Section 2, Anatomy of an enzyme. How do you actually build this ultimate biological machine?
It turns out it takes 4 distinct architectural steps. It kicks off with the primary structure. This is just the linear sequence of amino acids linked by peptide bonds. Basically the 1D blueprint. Step 2 is the secondary structure, where hydrogen bonds create these local folds, curling into spiral alpha helices or flattening out into beta sheets, kind of like molecular origami. Then comes step 3, the tertiary structure. Hydrogen, ionic, and disulfide bonds lock everything into a highly complex, functional 3D shape. And finally, some enzymes take it even further to a quaternary structure, linking multiple proteins subunits together. Lactate dehydrogenase is a great example of this.
But here's the catch. Even perfectly folded enzymes sometimes need a little help to get going. These chemical allies fall into 2 main camps. First up, cofactors. These are non-protein metal ions like magnesium, zinc, or iron. Then you've got coenzymes, which are organic helper molecules like NAD+, FAD, or coenzyme A. And fun fact, a lot of these crucial coenzymes are actually derived straight from the vitamins in your daily diet.
Next up, Section 3, The Perfect Fit. Let's look at how enzymes actually interact with their targets.
So the actual business end of an enzyme is this highly specific pocket called the active site. That's where the catalytic magic happens. Now, what's really fascinating is how our understanding of this site has evolved. Back in the day, Emil Fischer proposed the lock and key model, where the active site had this rigid, fixed shape and the substrate just clicked into it perfectly. But today, the consensus is Daniel Koshland's induced-fit model. This tells us enzymes are dynamic. They actually shift and change shape when the substrate binds, literally hugging the target to create the absolute perfect conditions for the reaction.
To make this a bit more concrete, let's look at what these structural classes actually do for you every day. DNA polymerase? That's busy replicating your DNA for cellular growth. When you grab a bite to eat, protease goes to work breaking down proteins, amylase tackles starches, and lipase handles the fats. These incredibly specialized machines are just constantly humming in the background, locking onto their substrates to keep your body running.
Which brings us to Section 4, when enzymes break down. We're looking at conditions, inhibitors, and diseases.
You see, these machines are incredibly sensitive to their environment. Take pH levels, for instance. Every enzyme has an optimal pH where its structure is perfectly aligned. Pepsin, for example, loves a superacidic environment with a pH around 2, which makes total sense since it lives in your stomach acid. Trypsin, on the other hand, operates down in your intestines and demands an alkaline environment, right around pH 8. If you swapped them, they'd be completely useless.
And temperature is just as critical. Sure, enzyme activity speeds up as things get warmer, but if it gets too hot, well, that's disastrous. High temperatures trigger what's called protein denaturation. Basically, the intense heat physically rips apart those delicate hydrogen and ionic bonds we talked about earlier. It literally melts the machine's 3D architecture, leading to a complete, irreversible loss of enzyme activity. Game over.
Beyond temperature, these structures also face molecular villains called inhibitors. In competitive inhibition, it's straight-up chemical combat. A rogue molecule literally fights the substrate for the active site, physically blocking the way. But then there's non-competitive inhibition, which is more like remote sabotage. The inhibitor sneaks in and binds to a totally different part of the enzyme. This warps the shape of the active site from afar, meaning the substrate can't fit anymore. And when these microscopic machines break down or suffer structural defects, the ripple effects on the human body can be severe. A defect in just a single enzyme, phenylalanine hydroxylase, causes the genetic disorder PKU. Low activity of the lactase enzyme, that's what gives you lactose intolerance. And tragically, defective lysosomal enzymes are what cause Tay-Sachs disease. It's wild to think that the failure of just one tiny biological machine can so profoundly impact a person's life.
All right, Section 5, the future of biotechnology, going from nature's tools to human engineering.
Because here is the crucial point. We aren't just studying these molecules anymore. We're actually harnessing them to reshape the world. We're using CRISPR-associated enzymes for pinpoint genetic engineering. We use them for massive biofuel production. The textile industry uses cellulases to make your clothes. And in medicine, we're developing enzyme replacement therapies to directly treat the very genetic diseases we were just talking about.
We've officially entered the era of enzyme engineering. Scientists aren't just foraging for enzymes in nature, they're actively upgrading them. Using methods like directed evolution, complex protein engineering, and advanced computational design, researchers are purposefully modifying these machines. They're boosting their stability, tightening their specificity, and supercharging their industrial performance to create totally new, sustainable solutions.
And the tools are only getting sharper. Right now, artificial intelligence is helping scientists predict insanely complex protein structures and even design entirely new, never-before-seen enzymes from scratch, including ones that can literally eat and degrade plastic pollution. We are finally learning to speak the structural language of life. So I want to leave you with this thought. As our computational power fully merges with biological reality, what incredible biological machine will we build next? Thanks for tuning into this explainer, and keep learning.